Hi everyone, my name is Ashley Valentina Schwartz, and today I'm going to be talking to you about how machine learning identifies the chemical properties that predict pancreas toxicity in the zebrafish model. A little bit of quick background on this project. Environmental pollutants can pose a hazard to embryonic and fetal development. So what we're interested in is how does chemical exposure affect pancreatic embryogenesis and organogenesis specifically. This is really important because alterations to pancreatic development during the embryonic stage could manifest disease later in life, such as diabetes. So to study this, we used the zebrafish model. They're a vertebrate model that closely mimic human embryonic development. As you can see on the image on the screen, they're completely translucent, which means we can visualize the organs develop in vivo. And even greater than 70% of human genes have homologs in zebrafish, which make them a really great candidate to study the gene expression changes. So our objective is to determine the impact of toxin exposure on pancreatic organogenesis by studying the transcriptomic response of the zebrafish to a variety of different chemicals. And the transcriptomic response is really using RNA sequencing to get an unbiased snapshot of the gene expression profiles following chemical exposure during that embryonic stage. We have done this before for a variety of different chemicals, and our study and a variety of studies ran across the country input their data and their results into what's called the Gene Expression Omnibus, which is a public data repository for gene expression profiles. A big limitation to a lot of these studies, including ours, is it is a lot less common to study more than one chemical. So my goal is to really leverage the vast amount of publicly available embryonic toxicity datasets that already exist to compare the transcriptomic response to a variety of different chemicals to learn more and hopefully build more predictive models in the future. A big challenge to this is not all studies are created equal, and there's a lot of data to deal with. So two big tasks that we have to tackle are data engineering and data analysis, which I will go into depth in just a second. So the first step is data engineering, and again, the challenge here is that all data is not created equal. And what I mean by that is that when studies go from raw sequencing reads to differential gene expression, there are a variety of different routes to take. They could use different softwares, and really the solution to this would be to create a normalized database by uniformly analyzing raw sequencing data for all the studies included in the study. And to do this, we have to have a lot of computational power. So we're using the SDSU Computational Science Research Center High Performance Computing Cluster to go from raw sequencing data that we collect from the Gene Expression Omnibus all the way to differential gene expression. Today, I'm going to be presenting on a case study dataset of 33 different chemicals that have been normalized for bioinformatics pipelines and even experimentation pipelines. And this includes 10 flame retarding chemicals, 1 TCDD, and 22 polycyclic aromatic hydrocarbons. The next step is data analysis. So the challenge here again is there is a lot of data. When we're starting with a transcriptomic response to 33 different chemicals, that means we're looking at the total gene expression profiles of over 30,000 genes per chemical included in the data set. And as we start to scale up the amount of chemicals that we're dealing with, you can imagine that number is going to get greater and greater. So we're starting with that transcriptomic response to 33 chemicals, and we're using a machine learning algorithm, specifically a hierarchical agglomerate clustering algorithm with word linkage. And from that, we're going to get the clusters of chemicals with a similar response for pancreatic gene ontology. You might ask, why did I choose this machine learning algorithm? Well, this is a big choice, and I go into depth on this in my poster. So please, if you have questions about that, come and talk to me there. It turns out the choice of clustering algorithm has a big impact in that final result, which is really important to identify for studies that use this sorts of methods in the future. So once we execute that clustering algorithm, we end up with 6 distinct clusters as identified by the colors on the left-hand side of chemicals that are identified on the right-hand side with respect to their genes involved specifically in pancreas development. And the heat map here represents the over or under expression of those genes involved in pancreatic development. What we found is that the chemicals were actually clustering based on their chemical property. So for example, cluster 1 was primarily phosphate groups and flame retardant chemicals. Clusters 3, 4, and 5 were mainly polycyclic aromatic hydrocarbons or PAHs. Now these are both chemical classes, but we found that even within these clusters 3, 4, and 5, chemical properties were identifying those clusters even further. So cluster 5 was primarily aryl hydrocarbon receptor activators. Cluster 3 were non-AHR activators and chemicals with low molecular weights and low bioconcentration factors. So ultimately what we found is that chemical properties and disposition of PAHs may play a role in pancreatic development. This is really important as we start to build predictive models of how chemical properties may be able to predict pancreatic developmental toxicity when we're looking at gene expression. And again, this is important to identify the manifestation of diseases that may be occurring later on in life. And again, just some concluding remarks here for a big picture. Machine learning reveals transcriptomic response similarities and differences, even within a chemical class. This is really important as we start to build predictive models based on chemical property so we can identify the developmental toxicity of a chemical without having to run any experiments. If you have any questions, please feel free to come to my poster or reach out to me by email. Thank you so much.